Welcome everyone for the ISB. Just very, very briefly, in case this is the first time you're joining us in the ISB Academy. So the ISB Academy is part of the International Society for Computation Biology. And what we aim to do is provide talks, seminars, address the latest advances that we have in the field of computation biology. We do run different series. We used to have the COVID one, not running anymore. We do have the indigenous voices. We have the most popular one, which is the webinar, a series in conjunction with the COSI, and the COSI is the Community of Special Interest inside the ICB. And then we have this tutorial series where we try to provide a really practical sort of tutorial. We started this a year ago, and what we're trying to do, we have all of our materials inside GitHub and Zenodo, and I'm going to go through that in a bit. But before I do, just some housekeeping. We do have a code of conduct, and code of conduct is a way we're trying to make sure this space is safe for everyone. So if you did witness any sort of unacceptable behavior, please do report it to the ISB Academy Code of Conduct with this email. I just want to try to say that what we're trying to do is making our training materials as fair as possible. And by fair, I mean findable, accessible, interoperable, and reusable. So we're trying to provide DOI or unique IDs for any talk that's given under CC BY license. We do provide metadata. We try to make some of these materials contributor, how do you say, like welcome and contributor, and easy to contribute to it by adding it to GitHub. And we try to make it also usable for future trainers and trainees. So yeah, if you do have any sort of feedback about the materials, just let us know and how we can make it easier for you. As I mentioned, we have all of our materials, or most of it, in GitHub organization account for the ISB Academy. I've just added, I think, the link in the chat, and I can add it again. And you're going to see also a repository for today's workshop where you can see some of the materials that we're going to present today. You're very welcome to comment, to go to this repository, open issues, ask about anything that is not clear for you, or also reuse some of these materials. We add most of them under CC BY license. And just to explain what CC BY means, is it means anyone is very welcome to reuse the materials, but they have to attribute the trainer or the one who created the materials. And you can see how you can attribute them inside the repository itself. The other thing that we do, so inside GitHub, we add codes mostly, but inside Zenodo, which is a general purpose repository, we add talks that have been given. So any talk that's been given as part of one of these tutorials, we add it to Zenodo. We cross-link it inside GitHub. But the reason why we add it to Zenodo, because Zenodo enable you to create DOI, or resistant identifier, which make these materials more citable, and it does provide some sort of matrix as well for the trainer around how many people have used these talks, downloaded or reused it. So please, if you do go to Zenodo, I can provide the link and use any of these materials. Please do make sure that you cite the one who gave these talk or provided these materials, and reference them in any sort of material that you've used. And yeah, I'm really very, very excited for today's. We're going to be learning about example genome browser with the REST API, and how to retrieve genome annotation data by Alina. Very, very excited for this. And thank you so much, Alina, for preparing the workshop and tutorial. I'm going to stop sharing and hand it to Alina so she can introduce herself and the materials.
Hello, everyone, and thank you for being on time and joining us for today's workshop. My name is Alina Mushtaq-Stolworthy, and I will be delivering the Ensembl workshop for today. So I am part of the outreach team at Ensembl, and we are a team of about 3 people who are involved in giving workshops such as today, and then also REST API workshops that will be tomorrow. And we're also the point of contact between the Ensemble users and our developers. And then we're also sort of working on various different things within the Ensemble project, such as the new website and documentation pages on the websites and etc. So let me just go on to my presentation. The agenda for today's course is that I will be talking to you about what Ensemble is. So sort of like a background information and introduction to an Ensemble, to the Ensemble Genome browser. And then we'll also be looking at how you can find a specific region and look at it in detail in the browser on various different genomes. I will be mostly focusing on human. Then we'll also talk about genes and transcripts in Ensemble, and also how you can export various different data types with Biomark. Tomorrow, which is part 2 of the course, there will be an overview of Ensemble REST API. So REST API is a language agnostic tool that you can use to access Ensemble's data. So if you're working with Python, R, Perl, Java, whatever language you're using, you can use REST API with that. And then I'll also be talking about how you can script around REST API calls. So the way that the workshop is going to work is that I am going to give you a presentation to talk about the different data types that we have and how we produce and process certain types of data. And then I'm going to move on to the Ensemble Genome browser, and we're going to see where you can find these different data types that I've been speaking about. And then after that, you're going to have some time for exercises. So these exercises are definitely not a test. So you don't need to provide me with any answers. These are just for you to go beyond the demo a little bit and try the Ensemble Genome browser on your own. So Batul has already shared the training page with you, but if you go to training.ensemble.org, so you can see this on my screen as well. But I think I'll also share the link for this in the chat so you can navigate to it easily. I hope everyone can see this. So once you're on training.ensemble.org, on the blue navigation bar at the top of the page, if you click on training materials, and then from here, we can just click on ISCB Academy. So this is the specific page for today and tomorrow's workshop. From here, you can find a PDF version of my presentation. So this will include the presentation from today and tomorrow. And then we have, if you scroll down the page a little bit, we have these different tabs over here. So these are the different modules that we're going to cover today. So to start with, we have species and genome assemblies. And if I open this, within it, we have 2 further tabs. So if I click on demo, this is everything that I will be going through in the demonstration after the presentation. So everything that I will be showing you on the Ensemble pages. So feel free to follow me along on the screen as I go through the browser, or if you want, you can just follow these screenshots and then do it on your own machine at your own speed. Underneath the demo, we have another tab for exercises. So all the way down, we have this tab for this module for species and genome assemblies exercises. And this is where you can find all of the exercises for this specific module. So again, the exercises are not a test. If you need help, definitely do let me know. Since I'm here, I'll be happy to go through the exercise with you. If not, then you do have the text answer and the video answer available for every exercise that we are going to be covering today. I'm going to scroll back up, and another link that I would like to point you out to is this link over here that says Living Document. So this, if I click on it, is a shared Google document for today and tomorrow. So we have some general housekeeping over here, some useful links, golden rules for online training, resources, and then we have the space for questions. So I find usually that with Zoom, and I think maybe with this platform as well, sometimes if you ask questions and if there's several people asking questions, I might lose the question because there's normally not the option of threading. But if you ask the question over here, then I can come back to it, or other people can add to it, or we can sort of like have a discussion on this here as well. This document is going to be available indefinitely, so you can always use it as sort of like a knowledge database as well. If you want, you can also ask questions over here anonymously if you're a little bit shy to ask questions. Again, there's no silly question. There's no, there's no pointless questions. So if you have at any point, if you would like me to repeat myself, or if you have a question about something that I'm talking about, then definitely please do let me know, either on the chat or preferably on this Google document over here, because then I can monitor this Google document throughout the course today and tomorrow. With this, are there any questions so far about everything that I've shown you? Okay, so if there are no questions, then we're going to move on to our presentation. Okay. Batul, would you please be able to confirm if you can see my screen and if you can hear me?
Yeah, I can hear you and see the screen. Perfect, thank you.
Perfect. So again, as I mentioned, you can find the course material on training.ensemble.org, which I've just shared with you. Over here, you can find the presentation, the demos, and the exercises that we're going to be doing today. So the objectives for today's course are to basically learn about what Ensemble is, what type of data you can get in Ensemble, how to navigate the Ensemble Genome Browser, and where you can go for help and documentations, as there is a lot of it available. So I'm going to start with a little bit of a history. So the first genome was sequenced by Fred Sanger and his team in 1977, and this was a small bacteriophage genome of about 5,000 bases. And this is what paved the way for sequencing of more complex organisms, including the draft for the human genome, which was published in early 2000s. It was a much larger genome of about 3 billion bases. So you can see that from 1977, this bacteriophage genome was about just 5,000 bases. And then we had a huge jump in early 2000s to this human genome, which is 3 billion bases. So there was quite an advancement in sequencing technologies. And since then, many other organisms have been sequenced as well. So the advances in sequencing technology has enabled the scientific community to sequence many different organisms, and now we have large consortia that are aiming to sequence all living organisms on Earth. But it has also enabled the sequencing of many different individuals of a given species. So for example, different people of various ethnicities to assess the genetic diversity within the population and allele frequency. Ensemble was started in 1999 when the human genome was in commission, as it was known that the automatic annotation would be essential. So Ensemble's goal was to automatically annotate the human genome and integrate this annotation with other biological data and make it all publicly available. And since Ensemble's launch in 2000s, many other genomes have been added to Ensemble. So why do we need genome browsers like Ensemble? Genomes are big and they contain a lot of data, and we're producing them at a fairly massive rate. And if you look at the sequencing data, it can look a bit like this, which is just a string of letters of genetic code, and this data is completely meaningless without annotation. The only way this data will mean something is if we work with it and we process it, which is what genome browsers do. So Ensemble is a type of genome browser that takes publicly available sequencing data from genomes It's important to remember we don't do any sequencing ourselves. We take publicly available sequencing data and then we make sense of that data. So we annotate genomes with genomic features like genes, gene variants, enhancers, and promoters, and we also perform analysis on this data. Ensembl is also called an added value resource, which means that it brings together information from a wide range of other databases in a single site and provides easy access to all of this data. We are not the only genome browser, so we also have a sister page called Ensembl Genomes. So this is dedicated to non-vertebrates. So the main Ensembl site, which is what I'm going to be focusing on, is based on vertebrates, whereas Ensembl Genomes is based on non-vertebrates. So in Ensembl Genomes, you can find taxa such as plants, bacteria, fungi, metazoa, and protists. Then you might have also come across UCSC Genome Browser and also NCBI Genome Data Viewer. We have lots of data in Ensembl. So genome assemblies are the primary data type that we import from publicly available databases, and then we annotate these genomes with genes, which are called gene builds. And we have gene builds for over 300 vertebrate species in Ensembl. We have genetic variation data, not for every species, but for a very good representation of them. We have comparative genomics that is calculated in-house, including genome alignments, gene trees, and homologs. And we also have regulatory builds for a set of regulatory elements, which are depicted from experimental data for human and mouse. We also have tools that help you access the Ensembl data. So we have Biomart, which is what we're going to talk about today, later on, which is an online export data tool. And this allows you to export large amounts of Ensembl data tables without any programming knowledge. We also have VEP, which stands for Variant Effect Predictor, to annotate your own variants. We also allow you to display your own data in the browser, and we also provide programmatic access to our data via our APIs. So we will be talking about REST API tomorrow. Most importantly, Ensembl is completely open source, so it's free to use, and we provide access to our whole genome files that are stored in our FTP site and GitHub. So we've already just hinted that there are different access scales. So you can access Ensembl's data one by one by using the browser, which is what we're going to do today. But you can also access our data in bulk using tools such as Biomart, Ensembl Web, and also our APIs and directly via MySQL. As I mentioned, we also have our FTP site. So this is a public directory with whole genome flat files, and these are basically large files if you wanted to download genome-wide data. As I mentioned before, that Ensembl, the main site, is dedicated to vertebrates. And there are about 300 vertebrates, and the number is constantly growing. But in the main site, you can also find a few non-vertebrate species. So you can find Drosophila, yeast, and C. elegans. And these are there because they are the most commonly used model organisms, but they're also there for historical and some comparative purposes as well. And then we have our sister site, which is Ensembl Genomes, and this is dedicated to non-vertebrates. So within this site, you can find Ensembl bacteria, protists, fungi, metazoa, and plants. These sites are organized in the same order as the main Ensembl site. It's just that the colors will be different, and you can access them by going to www.ensemblgenomes.org. And here I have a comparison between Ensembl and Ensembl Genomes. So as you can see that Ensembl is a little bit older. It was published in 2000s, and then Ensembl Genomes was published a bit later in 2009. So Ensembl's data is updated every 3 months as genomic data constantly changes. So our data is not static. So every 3 months, we release a new version. And behind the scenes, there is a lot of work as new genome assemblies are input. We update our gene sets, we update our software. We also run new analysis on all of this data. We update our variation and regulation data. And finally, we package it all up and display it in the browser. The current Ensembl release is 110, and for Ensembl genomes, it's 57. So this was released in summer this year, but this means that if you were to come back to Ensembl, let's say in January next year, or maybe like in a few months' time, the data there might not be the same. Therefore, it's crucial to know which version of Ensembl you're working with. And if you wanted to make your data consistent and you wanted to find the version that you were using in the past, you can do so with our archives. We store all of our versions in our archives, which keep frozen in time for up to 5 years. So let's talk about genome assemblies now. This is the primary data type that you can find in Ensembl, and we'll talk about how genome assemblies were generated in the past. So I'm showing you a bit of a historical approach over here on my slide on how the first human genome was assembled. So I will say here that currently there is no sequencing technique which can or is able to read the entire chromosome. Therefore, the DNA first has to be fragmented and the fragments are then sequenced. So back in the day, step one was to cut the DNA into chunks, and then this DNA was cloned into BACs. So these are bacterial artificial chromosomes, which were then grown in bacterial cultures to amplify the number of DNA copies. And finally, the cloned DNA was then sequenced from the BACs using Sanger sequencing. Later on, there was an era of PCR amplification, and nowadays, often you don't really need any amplification at all. So sequencing would then create these so-called reads, so-called sequencing reads of varying lengths, depending on the technology used. But in all cases, there is shortening of the genome, which is then matched up using overlaps to create a continuous sequence called a contig. And here on my slide, I have a bit of a terminology that is associated with genome assembly. So the set of sequenced BAC clones with overlaps plotted along the genome is called a tile path. So here we have the tile path, and each of those blue boxes that you see is sequenced BAC clones, and you can see that there's some overlap between them, and they're organized in order, so they are plotted along the genome. And then the BAC clones with trimmed overlaps are called contigs. And contigs are shown here as these blocks of different shades of blue. And then a set of contigs stuck together with no gaps is called a scaffold. And scaffolds would then be joined together to form a chromosome. I need to stress here that the human reference genome is not a genome of a single person, but at the same time, it's not a consensus genome either. So it is composed of chunks, and every chunk is a real person's sequence. So it's somehow a hybrid genome of different people's DNA. And people often assume that the genome assembly is a perfect representation of the genome. But I'm going to use this map analogy to show you what it's really like. So if you imagine the genome, or all of the DNA within the cell that corresponds to a map, this map is then going to have a coordinate system with some annotation and labels. And we would like this genome assembly to be as accurate a representation of the genome as possible. So something like you see in Google Maps. However, in reality, it's something more like this. So it's not perfect. There are gaps and there are errors, and there are some genomic regions that are difficult to sequence and reconstruct. There are some repetitive information as well. So with time, the scientific community is filling in these gaps and correcting errors, and the scientific community might issue some patches and corrections. So the bottom line is that the genome assemblies do change, and they're constantly being improved upon. And then this brings me to the versions of the human genome assemblies. So in Ensembl, we are currently using the latest Human Genome Assembly, which is called GRCH38, which has got fewer gaps in terms of base pair length, but also many rare and private alleles, which were previously found in the human reference, have been replaced with more common alleles. And our GRCH38 data and software are constantly being updated. We also have a site that is dedicated to the previous genome assembly, which is GRCH37. This is still widely used in the clinical community, but please bear in mind that it has got some larger gaps, and also our GRCH37 software is not regularly updated. Finally, we also host the even older Human Genome Assembly, which is NCBI 36. However, it is quite imperfect. There's lots of gaps, and the data, our software, are no longer updated. But it is still frozen in time, so you can find it in our archives. And I will show you where you can find the archives when we move on to the demo. So we're now going to move on to the Ensembl homepage, and we're going to look at how you can find information about different species that you might be interested in, and also the genome assemblies in Ensembl. So I'm going to jump out of my presentation, and I will be going to www.ensembl.org. So I'll type it in the chat as well. So it's www.ensembl.org. And this is going to take you to the homepage. So on the homepage, the first thing that we've got is this blue navigation bar at the top of the page. So here it shows you some shortcuts to our most popular tools. In this downloads link, you can find the link to our FTP sites. We have help and documentations to help you through navigating the Ensembl pages. And then we also have a link to our blog over here, where you can find articles on our new releases, so any updates that are being brought about in Ensembl. Then on the right-hand side, we have this summary over here about the data that Ensembl has, and also the release version, which is 110, and it was published in July 2023. We have some bullet points on the updates that were brought about with the release. And then we have, if you scroll all the way down, we have this tiny link over here that says View in Archive Site. And if I click on this, you can find all of our previous versions of Ensembl that are stored in time for up to 5 years. And you can also find a link to Ensembl GRCH37, which is the previous Human Genome Assembly. I'm not going to go to any of the archives. I'm just going to jump out of this presentation, out of this menu. Then if I scroll back up, you'll notice that we have 2 search options on this page. So anything that you search over here using this search option on the right-hand side at the top is going to give you results across all species. So if you were to search over here with a gene name, it's going to give you results across all species that we have in Ensembl. But if you would like your search to be species specific, then you can focus on the center search option. So from this drop-down menu, you can choose the species that you're interested in. So for example, I can choose human. And then if you type in your gene name or your coordinates, phenotype, anything that you search now is going to be specific to human. Then underneath this search option, we've got this genomes section over here. We've got our favorite genomes. They're set to human, mouse, and zebrafish by default, but you can click on this pen icon and you can change the favorite genomes over here if you want. To edit this, you will need to create an Ensembl account, but it's completely free to create and you do not get any correspondence from us. If you would like to view the full list of all the species that we have in Ensembl, so these are all the vertebrate species, you can click on View Full List of All Species just over here. And then it's going to show you a table with all of the species that we have. So in the first column, we have the common name, then we have the scientific name, we have the taxon ID, we have the Ensembl assembly, the accession, the gene build method, and then indication on whether or not we have variation and regulation data available for that species. You can filter this table So you can sort of like search for this table by typing in your species name or taxon ID or whatever. Or you also have the option to export this table by clicking on this Excel icon on the right-hand side at the top of the table. So it's just over here. I'm going to filter this table by human. And you can see that my table is now filtered to show me the human species just over here. So you can see we have the common name, Homo sapiens, the scientific name, we have the taxon ID, the current assembly that's being used in Ensembl, which is GRCH38, the accession number, the gene build method, and then whether or not we have variation and regulation data available for it. So Y stands for yes, indicating yes, we have the variation data, and yes, we have the regulation data. So I'm going to find out more information about the human species, and I'm going to click on human. And this takes me to the human species specific page. Again, anything that you search over here using this search option is going to give you results that are going to be human specific. Then we have some example pages over here for genome assembly, comparative genomics, regulation, annotation, and variation. If I focus on this section over here that says genome assembly, you can see that we have our genome assembly name that's being used and also the accession number. You can also download the DNA sequence in FASTA format. You can convert your data to GRCH38 coordinates. You can also display your data in Ensembl. And if you would like to find out more information about this genome assembly, you can click on more information and statistics. And this is what I'm going to do. So I'm going to click on more information and statistics. And this takes me to a page that gives me more information on this human genome assembly and gene annotation. So you'll see that we have some information here on assembly, patches, and annotation method. And then on the right-hand side, we have some summary statistics in a table format that give you information on the base pair length. So that's the assembly length, assembly provider, annotation method, and also we have information on gene counts. So we have our coding genes, non-coding genes, pseudogenes, the number of total transcripts, and so on. We also have information over here about the alternative sequence. I think what I would like to mention over here for that is so this is basically the assembly that's going to contain the haplotypes as well. So these are different versions of the same sequence. And then it's going to also include the total number of coding genes for that and non-coding genes, pseudogenes, and so on. So, yep, I think this is everything that I want to show you on this page. You can read a little bit more about the alternative assembly over here. So just on this left-hand side, and you can watch a video on YouTube as well about patches and haplotypes in the human genome. If there are any questions regarding this, please do let me know. And with this, I'm coming to the end of my demo. I'm going to go back to the homepage by clicking on this Ensembl logo. And are there any questions so far about anything that I've covered? If the species being uploaded doesn't have a known genome. What do you mean if you upload your own data onto Ensembl and that genome isn't available? If you could just expand on this question, then I'll get back to it. We'll have some time for exercises now. So we're going to do the exercises for species and genome assemblies. So these exercises are available on your training page, and I'll share the link for this training page again. So we've got an exercise on the panda species, zebrafish, mosquito, and then Ensembl genomes and bacteria as well. So you can try the one that's most relevant to you. You don't have to try all of them, just try one or two, because there's always more exercises than there is time for. So I'll say I'll give about 15 minutes for you to do the exercises. And if there are any questions in the meantime, please do let me know on this Google document. All right, how's everyone doing with the exercises? Are there any questions before we move on to the next module? Okay, so I will move on to the next module, but again, in the meantime, if there are any questions about any of the exercises that you've covered or anything that we've covered up to this point, please do let me know. So the next module is looking at a specific region in the human genome. So we'll be looking at, so we have our coordinates over here. And the way that these are formatted are in the Ensembl default format. So we have our chromosome number, which is 4. And then after the colon, we have our start coordinate hyphen the end coordinate. So I'm going to copy these. I'll paste them in the chat as well if you want to do them on your own machines. And I'm going to go on to the Ensembl Genome Browser. And from here, I'm going to paste my coordinates in the search option. So it's important to remember that genomic coordinates can be ambiguous. So we have to specify the species that we are referring to. So from this drop-down menu, I'm going to select human. So I've specified my species, I've specified the specific coordinates that I'm interested in, and now I'm going to click on Go to look at this specific region that we have searched for in the Ensembl Genome Browser. And this takes me to a tab, which is called the Location tab. So Ensembl's data is organized in a tab system, and the Location tab is the first tab that we're going to be exploring today. But as the day goes on, we'll also be looking at the Gene tab and the Transcript tab. I also would like to point out the page-specific help that we've got on this page. So on most of the Ensembl pages, you'll notice that we have these question mark icons that you can click to find out some page-specific help. So it gives you these nice screenshots and shows you what everything on the page means. So if you get stuck sometimes, or you're not sure that what the data is that you're looking at and you would like some explanation, then you can click on those question mark icons to find out more information. On the left-hand side, we also have these options for frequently asked questions, video tutorials, glossary, and also contact help desk. I'm going to close this help page, and we're going to go back to our Location tab. Then on the left-hand side, we have this list of available views in the menu, in this menu. So the menu is always page specific, and it lets you navigate through the page that you're looking at. And then below the menu, you can find that we have these blue buttons. So we first have configure this page button, which allows you to modify and customize the view, add more data, as by default, we show you a small amount of data. You can change the style of everything that you're looking at this page as well. Then we have custom tracks. So this allows you to upload your own data to the Ensembl Genome Browser to be visualized here on the Location tab. And finally, you can also export data from this view. So if you click on Export Data, it's this blue button on the left-hand side. You can select for your output format. So you can select it from FASTA, bed format, CSV, tab separated, and so on. You can choose your flanking region. And then if you click on Next, you can then also choose your file format from HTML, text, and compressed text. I'm going to close this window by either you can click on anywhere outside the window or save and close. Then our main view is composed of these 3 images. So you'll see we have this first image over here, then we have the second image, and then underneath it we have our third image. So our first image is our chromosome view. Our genomic region is indicated by this red box that's just over here. So this shows you where we currently are in the genome. We have different tracks that will be plotted along the genome. So tracks are basically a genome specific term. It basically just means data that is plotted along the genome. So in the track, you can see that we have our chromosome track over here with its cytogenic banding. And then underneath this image, we have our next image that's called region in detail view. So this is our image that is centered around our region of interest. So our region of interest is still in this red box. So it's basically, this image is sort of like a more zoomed in version of this first chromosome view, if that makes sense. So our region is shown in a bit more detail, so a bit more zoomed in. Again, it's in this red box, and then we also show you the region around the red box. So you can see that this image has a few more data tracks. So we can see a track here for contigs. So it's this blue line over here. It's this like different shades of blue that you've got. Then we have a track for genes. So genes are shown here as these blocks, and then their names will be aligned to the left-hand side of the gene, and the gene name is also going to have an arrow that is going to indicate the orientation of the gene. And then we have another track for regulatory builds. So these are basically all the regulatory elements such as enhancers and promoters that fall along the genome. You can find the legend for all of this just below these tracks, so just over here. Finally, we have our most detailed image at the bottom. So this is our third image, and this is our entire red box. So this is only our region of interest. So this is the region that we've specifically searched for by providing the coordinates. So you can see we have various different tracks over here, so lots more. As I mentioned before, that by default, we don't show you all of the data tracks that you can view. We only show you a few, and that's just so the page would load a bit more quicker. If we show you all of the data tracks, then it's going to take ages to load. But you can add different tracks to this image. And to do that, I'm going to click on Configure this page on the left-hand side. And from here, it's going to show you first all of the active tracks. So these are all of the tracks that are already turned on. And then on the left-hand side, we have this nice menu that is showing you different tracks in various different categories. So we have sequence and assembly, genes and transcripts, mRNA and protein alignments, variation, somatic mutation, regulation data tracks, comparative genomics data, and so on. So you can click on these various different categories and then choose your tracks, or you can also type in your track name over here where it says find a track and then search for your track. So I'm going to search for the UniProt track, and you'll see that this becomes available on this main section. So I have my search results over here. So I have a few search results. Some of them are from the mRNA and protein alignment category, and some of them are from the variation category. So you can choose whichever ones are relevant to you. To turn a track on, you can simply click on this box over here. And if I click on this, it's going to give me an option to choose my track style. So it's completely up to you, again, how you would like your track to appear. I'm just going to select normal. If you're not sure what a specific track means, then you can simply click on this eye icon in the same line as the track, and it's going to give you information on where this data track or where this data is coming from. So you can see that proteins mammal from UniProt data track are the mammalian proteins from the UniProt KB database that are positioned along the genome through BLASTP. Once you're happy with your track selection, obviously you can choose multiple tracks if you want. I'm only just going to choose one as an example. Once you're happy with your track selection, you can simply click on Save and Close. And then I'm going to wait for this data track to load. So all of your data tracks are going to appear in your third image, so your most detailed view. So you'll notice my data track for proteins from UniProt has appeared just over here at the top. So all of these features that you see on this page are interactive. So you can click on these features and it's going to tell you the UniProt match name. You can also find out information about the alignment length and so on. Also on this page, you'll notice that we have our blue line. So this is our contig. So the blue line is always our contig. So this is our reference genome. Anything above the reference genome is going to be forward stranded. And anything below the reference genome are going to be your reverse stranded genes, or the features are going to be reverse stranded. And I will talk about it more when we move on to genes and transcripts. There's lots of options to play around on this image. So again, if you click on this add remove tracks, this is basically the same menu that I showed you as configured this page. So you can use this to add more data tracks or remove certain data tracks, change styles, and so on. You can add custom tracks. You can share this page with collaborators. You can resize the image. You can export this image in various different formats. If you've added loads of different data tracks and you've manipulated lots of different things on here, and now you would like to go back to your default setting, you can click on Reset Configurations, and you can also reset track order. So all of these tracks that you see over here, you can change their order. So you can sort of click on them and drag them above or below, depending on your priorities. And then the really cool thing that we allow you to do is you also have the option to draw on this image. So if you select this dotted box over here, that means you have the option to draw. So now if I click on this image and drag my mouse across, I can select a specific region. And then I can either zoom in to that region or just mark that region. So that's quite a handy tool if you are going to sort of move around this image and then you'd like to come back to your marked region, then you can use this option. I'm not going to mark anything for now. So I'm just going to close this menu. And then you also have the option to scroll across this region. So now if I click on these arrows over here, so it's now changed the action of my mouse, and now I'm going to click and drag my mouse across. So it's going to sort of scroll across the region. Sorry, it's just loading at the moment. Whilst this loads, I'm just going to check if there are any questions on the chat. No, none, or in the document, none over here as well. Okay, so I'm going to assume that everything has been clear so far. So you can see that now I've scrolled across and I've sort of like moved more to the right-hand side of the image. Okay. So as you can see, the page is quite flexible. There's lots and lots of options for you to explore and play around with. So again, we're now going to have some time for the exercises. So the exercises, you can find that on the training page. And now we're going to do the next module, which is exploring genomic regions. So within exploring genomic regions, this demo, if you click on this, this is everything that I've covered just now. And these are the exercises that we're going to do. So the exercises are based on a genomic region in human. We have CRISPR site in human. We have assembly exceptions human, and also one based on rice. So that would be on suitable plants and also on salmonella. So you can choose one that you are most interested in. These exercises are going to take you a little bit more time, so I would advise you to only try and do one. And then let's say that we'll give about 15 minutes again. And if there are any questions, please do let me know. Okay, there are some thumbs up reactions coming in. So I'm assuming that you're happy for me to move on. All right, so it seems that the people who have responded are mostly ready to move on. So again, if there are any questions, please do leave them in the living document. Batul has already shared the link with you. And again, even if I don't get back to it right now, I will be constantly moderating the document today and tomorrow. So if you have any questions later on, then you can definitely type them here and I will get back to you. Okay, so I'm going to now move on to our third module, which is genes and transcripts. Let me just, sorry, I'm still familiarizing myself with the platform for today. What have I done? Okay. So on my slide, I'm showing you the region in detail view that you have explored in the demos and exercises for the introduction to Ensemble module. On this slide, we have different transcripts of a gene. And a gene is a collection of transcripts in Ensemble. So it's absolutely normal for many alternative transcripts to be transcribed from a single locus. They're also known as isoforms or splice variants. And personally, we're not a fan of the term splice variants, as that would imply that alternative splicing is the sole reason for alternative transcripts. However, it's more complicated than that. There's alternative transcription start sites and end sites. There's alternative translation initiation sites, and so on. All of the transcripts that you see are biologically valid and they are real. They could be expressed differently and they could serve different biological functions. So if you focus on the transcripts on the slide, the transcripts will have a specific biotype, which will be color coded. So we can distinguish between protein coding genes. So these are genes that produce proteins and are translated, and non-coding genes which do not result in a protein. So for example, like RNA genes. So protein coding gene can have protein coding transcripts which are translated, and these are shown in red. But protein coding genes might also have non-coding transcripts which do not get translated. So these, for example, could be retained introns, and over here this is shown in blue. Finally, we also have golden transcripts, and these are merged transcripts. So golden basically indicates higher quality and confidence of their annotation, and the golden transcripts can be coding or non-coding. And I will talk about golden transcripts more further on in my presentation. So what else can we learn from this image? Exons are shown as boxes. So empty exons are non-coding, which means they are not translated, they're only transcribed. Therefore, transcripts of a non-coding gene, for example, will be composed of empty boxes. Filled and colored exons are coding. So you can see that, for example, this red transcript over here has all colored boxes. So these are coding exons. You can also see that coding transcripts might also have empty boxes. So you can see it has this at this 5' end and 3' end. So these are UTRs, which are untranslated regions. So it's important to realize that not all exons are coding. UTRs are exonic, as they remain in the transcript after splicing, but they're non-coding as they're not translated. Then we have introns, and these are lines that connect the. Oops, sorry, too quick. These are lines that connect the exons, so they connect these boxes. There are a few hints indicating the gene orientation, so the strand on which the gene is located. The first hint is the relative gene position to the genome assembly. As you may have learned from the instruction video, that forward-stranded genes are shown above the genome assembly, And then the reverse stranded genes are shown below the genome assembly. We then have these little arrows that would be next to the gene names. So for example, this transcript name over here will have this arrow that's indicating in the forward direction. Whereas if you notice over here, for these transcripts, these are going to be pointing in the reverse direction. All right, so we use two different independent annotation methods, and both of them rely on real biological experimental data. The first is called the Ensembl automatic annotation, which is applied to every single vertebrate in Ensembl, and the other is called the Havana manual annotation, which is done by a group, so it's done by people, and it's done only for human and mouse, and then for some difficult regions, it's also done for zebrafish and rat. Whereas the automatic annotation is done for all of the species in Ensembl. The Ensembl automatic annotation is a genome-wide gene prediction using our automated pipeline, so it's based on real biological experimental data. For this, we take known proteins, transcripts, cDNA, RNA sequencing data, and we plot this along the genome using a sequencing matching technique. So people sometimes think that all we do is that we screen for open reading, we screen the genomes for open reading frames, but we do have to have real evidence that something is translated or transcribed to make that gene annotation. And it takes about a week or two to annotate a genome using our automatic pipeline. Here I have all of the sources for biological evidence that we use. So we take nucleotide data from from INSDC, which is International Nucleotide Sequence Database. And this is a collaboration with NCBI, DNA Data Bank of Japan, and ENA. We take most of our proteomics data from UniProt, that includes manually curated proteins from SwissProt and also some unreviewed translations from Treble. And then sometimes for rare cases, we might also annotate less studied or sampled organisms based on similarity to a better sampled organism. So for example, if there is not enough experimental data for a certain species, we might use data from a closely related species. So we might be able to predict, for example, genes in chimpanzee by mapping transcripts or proteins from human onto the chimp genome. Where this is done, we do indicate it on the page. Then we have manual annotation. So this is our second type of annotation, which is only done for human and mouse, and then for some difficult regions in zebrafish and rat. So Havana annotation is also genome-wide gene prediction on a case-by-case basis. This is done by a person, so an annotator, who moves along the genome utilizing the different types of data, including databases and papers. So it's more laborious. It takes about half a day to annotate a gene, and it might take up to several years to annotate just one genome. And this is why this is only done for humans and mice, because these are the most well-studied species. And again, for some difficult or specific genomic regions, it's also done in zebrafish and rat. The annotator can utilize a wider selection of evidence, including RNA sequencing data of both short and long reads, also data that supports transcript structures like CAGE and poly-A sequencing, which can tell us something about transcription start and end site. We use mass spectrometry protein data and publications as well. The advantages of manual annotation is that it's more comprehensive, especially for some difficult and tricky to annotate regions like non-coding features. It results in more genes in general and also more transcripts per gene. It results in more biotypes. It requires less evidence, so sometimes it is more sensitive. If there is small evidence, for example, then the automatic pipeline might just skip that evidence and not make any annotation. But the annotator, so the person that's working with the data, can explore that region further and can utilize other data types and make an informed decision based on case-by-case basis. So it's more accurate for some difficult regions like UTR sites, splice sites, assembly exceptions, and so on. The disadvantage is that it's slower. So it's laborious. And then to annotate one gene, I think it can take up to quite a bit of time. I think it's about months that it can take to annotate a gene. So you can imagine that it takes years to annotate the entire genome. And this brings me to the golden transcripts. So I did hint on the golden transcripts in my first slide, that these are merged transcripts. So now we know that we have these two independent annotation methods, the automatic and the Havana manual annotation, right? So the gene annotation for humans and mouse is a product. So this is an output of these two independent pipelines. So we take the two list of genes from the automatic pipeline and the Havana pipeline, and we merge them. And if there are gene models which are agreed upon between the two methods, then that transcript model is going to be given the golden color, which indicates the higher confidence and quality of these transcripts. And I had a question on GenCode in the living document. So it might be a tiny bit confusing, but GenCode project is simply the set of ensemble human and mouse genes. So it's called GenCode for historic reasons. But GenCode gene set is basically this merged set of human and mouse genes. So it's agreed upon between the automatic pipeline and the Havana manual annotation as well. And it's the default gene set that is used by many international projects that are listed on my slide. Another thing that I would like to mention whilst talking about transcript quality is the ensemble canonical transcript. So this is a single representative transcript that is identified at every locus. These are basically transcripts which are on balance. They are most highly conserved, most highly expressed. They are in concordant with other transcript scoring algorithms. They're also in concordant with UniProt canonical isoforms, and they have the longest coding sequence. And they cover the largest number of clinically important variants, and they have to be complete. So these transcripts have to be complete. And the canonical transcript flag is given to, as I mentioned, that it is a single representative transcript that is identified in every locus. And then we also have canonical transcript for all the species in ensemble. Then we also have main select, which is another project which stands for matched annotation between NCBI and EMBL API. So this is just another reassurance of transcript quality. So I mentioned that apart from us, there is another genome browser called NCBI, which have their own gene annotation and gene set, which is called RefSeq. So main transcripts are transcripts that are 100% identical between ensemble and RefSeq from NCBI. By 100% identical, we mean that there are a complete match from transcription start until the end, including both coding and non-coding parts of the transcript. So where available, ensemble canonical is going to match main select. Ensemble canonical is available for all the species in ensemble, whereas main select is only available for human. Then we also have main plus clinical. So very recently, the main project released an additional set called main plus clinical, and there's only 43 of them genome-wide. So in very rare occasions, there might be an additional main plus clinical transcript on top of the main select. In cases where there are some clinically important variants, so pathogenic variants which were not captured or covered by the main select transcript. So then main plus clinical was introduced to capture this variation. So they might not be the most representative, but they are very important for the clinically relevant variants. And finally, last two things that I would like to talk about with you is Ensembl stable IDs. So all of our features in Ensembl have assigned a unique Ensembl stable ID. These are composed of the ENS prefix, which stands for Ensembl. And then we have a letter indicating the type of feature. So G for gene, T for transcript, P for protein or peptide, and then E for exons. And this is followed by an 11-digit unique number. For non-human species, a suffix is added. So in case, for example, for mouse or mus musculus, we'll have ENS for Ensembl, mouse or moose, and then the letter indicating the feature followed by the 11-digit unique number. Again, for zebrafish, we will have ENS, DAR for Danureria, G for gene, followed by the 11-digit unique number. And you can find further information on this by referring to this link on my slide. And then the last thing I would like to talk about is gene ontology. So human language is ambiguous, and there are many synonyms or multiple terms for the same thing. And we might all be describing exactly the same thing, but using different language. It's the same in the scientific community. So for example, one person could be describing innate immune response, calling it innate immunity, and another person might be calling it non-specific immunity. We know that they both mean the same thing. However, computer searches don't cope with this very well. And that is why the scientific community has come up with these gene ontology terms, which is basically standardized language, which is easily searchable. It is controlled to talk about gene function to make sure that we're all talking about the same thing. And all of these terms have their accession number, they have the name, they have a type and a definition. Gene ontology terms are organized in hierarchy, which means that a given name, so for example, innate immune response, can have a more general parent term, and then also might have some child terms, and there could be quite a lot of them, as you can see on my slide. If you would like to find out more information about our annotation system, then I would recommend that you refer to this publication on my slide. And with this, we're going to now move on to the demo. So I'm going to be looking at the human gene called UQCRQ, and we're going to find out more information about it and its transcripts. I'm also going to type this gene name in the chat, UQCRQ. Okay. And then I am going to go to the Ensembl page by going to www.ensembl.org, or you can simply click on this Ensembl logo and you will be directed to the homepage. From here, I'm going to focus on the center search option. And from the drop-down menu, I'm going to select human, and then I'm going to type in my gene name, which is UQCRQ. Again, gene names can be ambiguous, so we can have UQCRQ occurring in mouse or zebrafish, so it's important to specify the species that you're interested in. Once you're happy, you can click on Go. And we have our search results over here. I'm going to click on the first search result, which says UQCRQ human gene. And this takes me to the Gene tab. So we looked at the Location tab previously, and now we're looking at the Gene tab. So the Gene tab is going to have information about my gene name. We have our Ensembl ID for this gene. We have a description, gene synonyms. So these are just names that this gene was previously known as. We have the location of this gene, so it's located on chromosome 5. We have the coordinates, and it's located on the forward strand. It just says it over here. We have further information about this gene that it has 6 transcripts, 207 orthologs, and it's associated with 3 phenotypes. To view the transcripts that are associated with this gene, we can click on Show Transcript Table. So it's this blue button. And now it's going to show me all 6 transcripts that are associated with this gene. So we have their ID, the name, their base pair length, their amino acid length, their biotype, their matches in CCTS, UniProt, and RefSeq, and also some flags for the transcripts. So you can see that I was talking about main select, ensemble canonical, and some other flags from GenCode and APRI. You can hover over these and find out what these different flags mean. I'm going to close this transcript table for now by clicking on Hide Transcript Table. We have a summary for this gene. So over here, we have information about the main select transcript for this gene, UniProt match, RefSeq match, ensemble version, where this gene maps to in other assemblies, so where this would map to in the previous human genome assembly, which is GRCH37, the gene type, so it is a protein coding gene, and so on. On the left-hand side, we'll have some gene-based displays. So you can explore this gene further. You can view the sequence of this gene. So this is what we're going to do. I'm going to click on sequence on the left-hand side. In here, we can see that we have our sequence of the UQCRQ gene. So you can download this sequence. You can blast this sequence. The exons of our UQCRQ gene are shown here in this brown letters. The sequence is given in the FASTA format. So basically all exons are highlighted in this peach coloring. So these are our neighboring exons just here. And then our exons of the UQCRQ gene are shown here in brown. You can also blast a selected sequence. If you were to click on the sequence and then drag your mouse across, you can select a specific sequence and then blast just that selected sequence. You can also configure this sequence if you want. So if you click on configure this page on the left-hand side, you can choose your 5' flanking sequence, your 3' flanking. You can choose the number of base pairs that you would like to view per row. You can show variants, for example. So all of the variations that overlap your sequence, you can choose from yes or yes, show links. So I'm going to select yes. And then if I save and close this menu, so I've clicked on show variants, yes. And now if I save and close this menu, it's now going to load or mark up all of the variants that span this sequence. Just going to wait for this to load. Sorry, it's taking a bit of time. Okay, I'm going to have to move on to the next page. This is just taking a bit of time, but hopefully when you try it, it'll work for you. But what you will see is different colorations and decorations on your sequence, highlighting all of the variants that fall within this region. The next thing that you can also look at is gene ontologies. So on the left-hand side of the page, we have ontologies over here divided by gene ontology, biological process, cellular component, and also molecular function if available. The variants have now loaded on my page. So you can see that we have all of these variants that are highlighted. You can click on these variants as well to find out more information about them. So if you click on them, it's going to give you the variant ID, the classification of the variant. So it's an indel, where it's imported from. So it's imported from dbSNP, the location of the variants, alleles that are associated with it, and so on. I'm going to close this menu, and I'm going to click on biological processes on the left-hand side. So this is on the left-hand side under ontologies. I'm going to click on biological process, and it shows me this really nice table that shows me all of the different biological processes, their accession number, term, evidence, and their association with the various different transcripts on this table. You can also filter this table, and you also have the option to download this table or export this table by clicking on this Excel icon. The last thing that I would like to show you on the Gene tab is external references. So also on the left-hand side, I'm going to click on external references. And this shows me a table with all of the external databases, identifiers that our gene corresponds to. So we have information over here from Expression Atlas, Gene Cards, HGNC symbol, MIM, NCBI match, and so on. So you can click on any of these links and it's going to take you to that database. So this is what the Gene tab looks like. And now we're going to look at the Transcript tab. So I'm going to show Transcript table. And from here, I'm going to click on this first transcript. I'm going to click on its ID. And this is going to take me to the Transcript tab. So you can see that the Transcript tab opens next to the Gene tab. For the Transcript tab, we have our Transcript ID. We have our transcript name, which is UQCRQ203. Again, we'll have the description of the transcript, the gene synonyms, the location for this transcript, and then about this transcript that it has 3 exons. It is annotated with 11 domains and features. It's associated with 802 variant alleles and maps to 289 oligoprobes. I'm going to hide the transcript table. Then underneath, we have a summary. So it shows you a really nice graphical representation of this transcript. So you can see that it has 3 exons. And then 2 of these exons are coding because 2 of these exons are colored in. And we have the same information given here in the statistics line. We have the transcript length, the translation length, and then number of exons, and also the coding exons. You can also explore this transcript tab further by focusing on this left-hand side menu. So we have, again, sequence over here on the left-hand side, and this is now divided into exons, cDNA, and proteins. So you can view the exon sequence or the cDNA sequence or the protein sequence. So if I click on exons on the left-hand side, it's going to open a page for me, giving me a table which shows me the exon ranking in the first column, the exon IDs, the start position of the exon, the end position, and also the length of these exons. And then in the last column, we have the sequence for our exons. So the sequence is colored differently. So green shows the flanking sequence. UTR is shown in orange. Intron will be shown in gray. And then your translated sequence is going to be shown in blue. So as you can see that this is quite busy at the moment, and that's because this exon sequence also highlights all of the variants that fall within the sequence, but I can remove the variants for now by clicking on Configure this page on the left-hand side. And you can sort of manipulate the sequence that you see by various different choices over here. But what I'm going to do is where it says show variants, I'm going to select no, just so that my sequence appears a bit neater for demonstration purposes. Once you're happy with that, you can save and close this menu. Okay, so you can see that my sequence appears a lot tidier. And as I was saying, flanking sequence is shown in green, UTR in orange, introns are shown in gray, and our translated region is shown in blue. We have a legend for it just at the top of the table over here as well. Again, you can also select a specific sequence and you can blast that selected sequence. And then you also have the option to download this sequence by clicking on this download button that's just above the table and the legend. In Ensembl, for the transcript tab, we also give you some protein information. So proteins are not going to have their own tab in Ensembl because protein features don't map to the reference genome directly. They map to the reference genome via transcripts. This is why we give you some protein information in the transcript tab. So in the protein information, you'll see that we have protein summary, domains and features, variants, PDB 3D protein model, and also AlphaFold predicted model. I'm going to click on protein summary. And this shows me a really nice protein summary for the protein domains for our translation. So we have information here from PFAM, Panther, Gene3D, plotted along the genome. And then you can also see some sequence SNPs over here. And we have the legend for it just below and some summary statistics just over here where it says statistics. You can also view the 3D structure for this translation. So if I click on PDB 3D protein model, so we have our PDB protein model over here. So this is quite an interactive widget that you can sort of play around with. You can turn it around. If I click on exons just over here on this eye, it's going to highlight the exons for me. So we have these 2 coding exons that are highlighted. You can also highlight your variants on this image. You also have the option to take a screenshot from this image, or you can also sort of manipulate it by clicking on show controls. Also on the left-hand side, you'll notice that we have information for genetic variation that you can explore in your own time. We have under external references, we have general identifiers. So these will be all the identifiers from other databases that match to this transcript. And also you can find information on microarrays from oligoprobes. So this is everything that I want to show you for now. It's a lot of information, and you can see that you can explore this transcript tab and the gene tab in various different displays. And I will give you some time to do that. And also we'll have some time for exercises. So the exercises that we're going to do now are for genes and transcript. So it's this 3rd module for today. And if I open this genes and transcripts exercises tab, you can see that we have one over here for MYH9 gene in human. We have this one on associated phenotypes, DPP6 gene in mouse, and also one on plants, so on Arabidopsis, if you're interested in that. So try one, and we'll give about, again, 15 to 20 minutes. And if there are any questions, please do let me know. Okay, so I hope that was enough time for you to try the exercises for genes and transcripts module. I'm going to move on to our last module for today, which is Biomart. Okay, so Biomart is a really cool tool that you can use to export data tables from Ensemble. It's highly customizable. It allows you to export large amounts of Ensemble data tables without any programming knowledge. All you have to do is build a few queries with a few mouse clicks, and you can generate custom data tables and sequence files. So Biomart is great for things that would be time consuming and difficult and tedious by using the website alone, as I've shown you just that there are different tabs that you can use. So for example, if you wanted sequences of 100 genes from your list, doing it one by one from the Ensemble Genome browser is going to take you ages. However, with Biomart, you can do it with a few mouse clicks. So it's great for things like ID conversions. As researchers, you often end up working with different databases, with all of them having their own identification system. So Biomart can convert IDs for you. Biomart can also export large amounts of data, but it does have some limited capacity. So Biomart is not a whole genome data export. If you want whole genome files, then please go to our FTP site, which is a public directory. If you ask Biomart to output all possible human genes, it's going to crash. So we would suggest around 5,000 items at one time. You can find Biomart on the blue navigation bar at the top of the Ensemble pages. So it'll be blue for Ensemble vertebrates, and then depending on the taxa, it'll be different for Ensemble genomes. To use Biomart, you have to follow 4 simple steps. So the first is that you need to choose your database and species of interest. You then have to narrow down this big database of all possible genes to a smaller data set of interest. Then you'll be applying, so you'll be doing this by applying filters, and then you'll be choosing your attributes. So these are things that you want to print in your output. So this is everything that you would like to appear in your results table. And finally, you can view your results and you get the option to export them. So if we look at the first step in a bit more detail, which is defining the data set, you can choose different databases depending on what you're querying for. You can look for database for Ensemble genes, variation, regulation, and mouse strains. So you're likely not to use mouse strains unless you're looking for specific differences between different mouse strains, and then you will need to define your species of interest. You'll then have to apply filters. So these are things that you want to search with. Most of the time, we'll be interested in a subset of genes that match certain gene names from our list or are associated with a given phenotype or function, or that are falling in specific regions. So Biomart gives you a lot of flexibility. You can apply one filter and build a simple query, or you can apply multiple filters to build a more complicated query. Biomart follows n-logic. This means that it gives you intersectional filters. So if I'm interested in all genes in chromosome 9, and then my second filter, for example, is all genes with a transmembrane domain, then in this case, Biomart will give you transmembrane domain genes on chromosome 9 as an output. Sometimes if you don't get results, this means that there was no overlap between your filters. Next, you will need to specify your attributes. So this is what you want in your output. And we provide a wide selection of these, including IDs, features, variants, homologs. These are orthologs and paralogs, and sequences. And then finally, you can view and export your data. So Biomart supports several file formats, including HTML, CSV, which is comma separated, tab separated, XLS, and FASTA. With XLS, just be mindful that if you are exporting your results as into Excel, then sometimes Excel likes to convert gene names into months or dates. So OCT2 as a gene name could be converted into October the 2nd. So just be mindful of that. If you are an R user, there's also an R package for Biomart, and you can find information for this by referring to the links on my slide. Okay, so that was the introduction to Biomart. And now we're going to get a bit more hands-on. So we are going to look at a set of 6 human genes. So I have these human genes mentioned over here in this blue letters. So we have gene names. but we would like to find out their NCBI IDs, their gene ontology terms, and also we would like the cDNA sequences for these 6 genes. So the first step is going to be choosing our data set, which will be ensemble genes, and we're interested in human genes, so we will say Homo sapiens. Next is filters. So we will need to narrow down the entire database of all human genes to match our filters. So these will be our gene names. So these gene names that are provided in this exercise. Then we have our attributes. So these are things that we would like in our output. So these would be NCBI gene IDs, gene ontology terms, cDNA sequences, and we will also include gene names over here just so that we can match our output to our input. And then finally, we can view and export our results. So let's try this in Biomart now. So I'm going to jump out of my presentation and I'm going to go to the Ensembl homepage, the Ensembl page, and you can see that I can find Biomart on the blue navigation bar at the top. So I'm just going to click on Biomart. So it opens Biomart for me, and this is what your page is going to look like. So the first thing that we will need to do is we will need to choose our database. So from this drop-down menu, you can choose from Ensembl genes, Ensembl mouse strains, Ensembl variation, and Ensembl regulation. 110 just means the version of Ensembl that we're currently on. We're interested in genes, so I'm going to select Ensembl genes. Then we'll have to choose our data set. So this is the species that we would like to work with. We're interested in human genes, so I'm going to select human genes from here. Once you've selected your data set, the option of filters and attributes is going to become available on the left-hand side. So I'm going to select filters first. And as you can see, you can filter by region, by gene, phenotype, gene ontology, and so on. I'm going to copy these gene names from my exercise. So I'll paste them in your chat as well. So you can give your gene names or your list as comma separated, tab separated. So let me just go back to Biomart now. So I'm going to expand gene by clicking on this plus icon. And as you can see that you can limit your entire database of all human genes by limiting to specific databases, by inputting external reference ID lists. This is what we're going to do in a minute. You can limit your genes by microarray probes, input specific microarray probes. You can limit by gene type, by transcript type, and so on. I'm going to select input external reference ID because we're going to provide these external gene names. And then in this text box, I'm going to paste in my gene names. I'm going to remove the and over here and separate them by comma. So from this drop-down menu, I'm also going to select the appropriate reference. So it's not gene stable ID, it is gene name. If you want, you can also choose a file from your computer and just upload a file. Again, the file, I think the names will have to be in a list or comma separated or tab separated. So you can see that we've applied our filter, and this also becomes available here on the left-hand side under filters. So it says that the filter that you've applied is gene names and ID list is specified. If you would like to make sure that your filter has been applied, you can click on count, which is just over here on the left-hand side. And this is going to show me that 6 out of 70,000 human genes match my filters. And that's true because we provided 6 gene names. The next thing is attributes. So these are things that we would like now in our results table. So if you remember that our exercise asked for, so our exercise provided 6 gene names, and it asked for matching NCBI IDs, gene ontology terms that are associated with those genes, and cDNA sequences for those genes. So we have these different categories for the attributes. So we have features, structures, homologs, germline variants, somatic variants, and sequences. You can only select one attribute category at a time. So I'm going to stay with features. And I'm going to expand this gene tab. And then from here, you can select everything that you would like in your output. So let's say we would like the gene stable ID, not the version. Let's say we would also like transcript stable ID, but not the transcript stable ID version. I'm going to select gene name over here just because I would like to match my output to my input. If I scroll further down, I'm going to select this external tab. And then from here, I have gene ontology information. So I'm going to select gene term, ontology term accession, term name, and term definition that is associated with the 6 gene names provided. And we would also like their NCBI IDs. So from external references over here, I'm going to look for NCBI ID, which is here. So now I'll also have the NCBI gene ID for the gene names that I've provided. All of the attributes that you have selected will become available on this left-hand side under attributes. Sorry about the Slack notifications. Okay. So now, once I'm happy with the attributes that I've selected, I'm now going to click on results. And it's going to show me a table showing me the gene stable ID, the transcript stable ID that matches the gene name that we've provided, the gene ontology information, and also the matching NCBI IDs. By default, Biomart will show you only the first 10 results. But if you would like to view all results, you can go to this view line, and then from here, select all, and it's going to open a new tab for you, and it's going to give you the results of all of your genes. So you can see that even though we provided just 6 gene names, the results here are the table is quite huge. And this is because it's giving you information on all of the transcripts that are associated with your genes as well. So if you remember that one gene can have multiple transcripts, so it's giving you the results for all of them. So I'm going to close this table, this tab for now. You can export your results in either by selecting file, or you can select compressed web file, and then you can provide your email address and email the results to yourself. The results can be, again, table separated, comma separated, HTML, or XLS. So we've got the gene ontology terms that match our gene name. We've got the NCBI IDs that match our gene name, but we still need the cDNA sequences for our gene names. For this, you don't need to create an entire new query. You can just go back to attributes by clicking on attributes on the left-hand side. And then from here, if you select sequences and then expand the sequence tab, you can choose from various different sequences. So we have peptides, coding sequence, cDNA sequence, exon sequence, and so on. I'm going to select cDNA sequence as that is what the exercise asked for. You can also choose, if you want, your upstream and downstream flanking size. And you can choose your header information as well. So you can choose what sort of information you would like in your header. So let's say we would like the gene stable ID. We would like the gene name. And we would also like the transcript stable ID, but not the version. Once you're happy with this, you can now click on results. And it's now going to give you all of your cDNA sequences for all of the transcripts of the 6 genes that you have provided. Again, Biomart is only going to give you the first 10 results, but if you want all of the results, you can go to this view line, and from this drop-down menu, select all. So you can see that we have our cDNA sequence over here, and in the header, we have our ensemble stable ID, the transcript stable ID, and also the gene name. And you can download these results in the FASTA format. Okay, so this is everything for today. So we have covered introduction to Ensembl. We have covered how you can explore specific regions in Ensembl. We've looked at the gene tab, the transcript tab, and now I've shown you how you can export these different data that I've shown you in Ensembl via Biomart. I'm going to have a quick wrap up, and then we're going to have some time for exercises and any questions that you might have. So Ensembl is a genome browser that integrates gene annotation, variation, comparative genomics, and regulation data. And I've shown you how you can access the Ensembl's data via browser and also via Biomart. Tomorrow, we will be covering how you can access Ensemble's data via REST API. So there are no questions on the living document. Okay, there is a question about, can different Ensembl stable IDs map to the same ENSTs? Yes. So as I was saying, that a gene in Ensembl is a collection of transcripts. So one gene can have more than one transcript. So it can have 6 or even 12 transcripts that are associated with it. That's why, yes, one ENSG could map to several different ENSTs. So T's is for transcript. I hope this answers your question. If there are any questions, please do let me know. We have still a little bit of time. So I'll give you about 15 minutes to check out the exercises that we have for Biomart. So these are on the training page in the Biomart tab. So please do go through them. If there are any questions, please do let me know. And then as this is the last module for today, after the 15 minutes time, I'm going to close the session. And hopefully I will see you all tomorrow as well. But I'll be here for the next 15 minutes whilst you're doing the exercises, or if there are any questions that you would like to discuss. So it's 6 o'clock now here in the UK, and I don't see any further questions on, well, there's a question coming on the living document, so I'll just wait for that. There are no questions here on the chat. Please do let me know how you're doing with the Biomart exercises. If you do them later, then you can definitely write down your questions in this Google document, and I will get back to them tomorrow as well. Okay. I'm sorry that you're having difficulty with sending the results to your email via Biomart. I'm not sure why it's doing that. I can have a look as well, but if you would be able to share a screenshot with me, so you can share the screenshot on this page. And actually, let me go on to Biomart and try this as well. So I'm just going to for example, just provide my email address here. And so for me, it says that the results are being compiled in the background, and an email will be sent to me when it's ready. So it seems to be working for me. I think you might want to like refresh your page, try again, or if it's still not working, then please can you share the screenshot over here, then I can have a look for you, and I'll probably get back to you by tomorrow. If there are no further questions, then I hope everybody has a nice rest of their day, and I will hopefully see you all tomorrow for REST API. Okay, thank you very much, and welcome everyone. Today, I'm going to be talking about Ensembl REST API. So let me just get onto my presentation. So the way that the workshop is going to work, again, it's going to have the structure of, I'll be giving you a presentation about the background and the theory and some code examples. And then we're going to have a demo where I'll show you where you can find the specific endpoints for queries that you're looking at. And then we'll have some time for exercises. So today's workshop is going to be a lot shorter than yesterday. So I'm aiming to finish by perhaps about 5:30 to 5:45. So again, the course materials can be found on training.ensemble.org. And let me just go ahead and open these for you. So just open them in a new tab. So if you go to training.ensemble.org, and then you can click on the training for today, which is just over here. So it's ISCB Academy using the Ensemble Genome Browser and REST API to retrieve genome annotation data. And we are going to be utilizing the Jupyter Notebook. So I have Jupyter Notebooks here for Python and R, and these are based on the human species. So feel free to use the ones that you are most comfortable with. So if your comfortable language is Python, then go ahead and choose Python, or if it's R, then you can choose R. So I'll be using Python. So I'll just show you what it looks like. So I'm going to open this in a new tab. And so these Colab notebooks, they are hosted in Google Colab. So to use them, you will need to have a Google account. And I think in this day and age, all of us have a Google account. But if you don't, then I'm afraid you might have to create one to use these notebooks. And further on, you will also need to go to File. And then you will need to say to save a copy in Drive. So I'll go ahead and do that. So you'll need to create a copy. And then once you've created that copy, this copy is now yours to keep. So it is yours indefinitely. You can play around with the code, change the code, and sort of like practice and learn from it. So these Google Notebooks, or these Jupyter Notebooks, they are interactive. So you can see that just as the exercises that we have on the training page, all of these exercises are going to have the exercises parts over here, and then you're also going to have a tab with all of the answers given. So all of these answers are hidden, but you can just click on the tab and then unhide them as well. And then we also have lots of example codes and sort of like all the demos that I'll be going through as well. So also, yesterday we also have our we had our Google document that you can use to ask questions. So again, please feel free to type your questions over here. I can get back to them either later on after I'm done with the presentation, or even like after the course, if there are any questions that are unanswered. You can also share your code over here and screenshot. So it just makes it a lot easier than using the ISCB platform that we're on, as there is no threading. So before I go on to presenting, are there any questions about the Jupyter Notebooks or anything else regarding the workshop? Okay, so I'm just going to have a quick recap of the Ensemble features. So Ensemble is a comprehensive database that is composed of different features, including genome assemblies and gene builds for over 300 vertebrates. We also have some comparative genomics analysis, which we can calculate in-house, including whole genome alignments and gene trees. We have variation data and web tool, allowing you to annotate your own variation data. We have regulation data on human and mouse, and then we also allow you to visualize your own data in Ensemble and plot it against the genome in the browser. Yesterday, we also discussed about Biomart, which is our online data export tool that allows you to export Ensemble data in bulk, and we also support programmatic access to our data via our APIs. We have Perl API and also REST API, which is language agnostic. Finally, Ensemble is open source, so it's open access and free to use. I mentioned yesterday about archives. So we know that Ensemble runs on this 3 month release cycle, and then the previous versions of Ensemble are stored in time for up to 5 years in our archive. So we have REST archives starting from release 87. The REST archives are for GRCH38 only, so the current Human Genome Assembly. And these are kept frozen in time for up to 5 years to match the Ensemble web archives. And to use the archives, you'll just have to use a slightly modified server. You just need to add the prefix. Over here, for example, over here it says e87.rest.ensemble.org. So our REST API is modeled around biology, which follows this parent-daughter relationship. So higher up, we have DNA, which is translated into pre-mRNA, which is then spliced by removing introns to give rise to mature mRNA, and then coding portion of the mRNA is translated into the protein. So the primary features in Ensemble are genes, transcripts, and exons. So pretty much anything which can be mapped to the genome directly is a feature. And then a gene is a set of alternatively spliced transcripts, and transcripts is a set of exons. So you can see the exons are shown here as these boxes. So a set of exons is a transcript, and then a collection of transcripts together is a gene. We also have translations, but these are not features in Ensemble as they do not map to the genome directly. They map to the genome via the transcripts, so they are a product of a transcript and they're not stored in the database. They are computed on the fly by the transcript object, and translations are defined by CDS, so coding sequence. And I would like to remind you that not all exons are coding. So exons, so for example, over here, you see this non-coding exon at the 5' UTR and this 3' region. So these are untranslated regions. Also, please remember that not all transcripts have translations. There are many non-coding transcripts as well, like RNA transcripts, which do not produce protein. Finally, all of our features have a defined location on the genome. So every feature, every object has a defined start and end position. And it's important that the start and the end of a feature is always defined on the forward strand, which means that the feature start and end might not reflect the transcription start and end. This is because the start coordinate always has to be smaller than the end coordinate in Ensemble. So we have two examples over here. So I have this gene SCNN1B, and there's this reverse stranded gene called PALB2. So we have forward stranded genes and reverse stranded gene over here, and the start and end are always defined on the forward strand, irrespective of the gene orientation. So for a forward stranded gene, the feature start would match the transcription start, and then the feature end would match the transcription end. But for example, for this gene over here, for this reverse stranded gene, the feature start would actually match the transcription end, and the feature so the feature start is going to match the transcription end, and then the transcription end is actually going to match the feature start. I hope this makes sense. So what is REST API? A REST API stands for Representational State Transfer, and it describes how one system can communicate with another. So if you imagine a client, for example, your computer in this case, trying to communicate with another. So imagine a client over here communicating with our server, which is Ensemble, over HTTP method, using this language agnostic method to access some of our data or our services. So for example, your computer sends a URL, which is, I've made it up over here. I've called it rest.ensemble.org/data I want. So it's specifying data you want, which is made up. So our server is going to interpret this, and it's going to return the requested data in a specific format. So Ensemble's REST API is language agnostic, which means that it provides access to Ensemble data independent of any specific programming language. So you can see that you can use many different languages to access our data using the REST API. However, it only provides a fraction of the Perl API functionality. Nevertheless, it seems to be sufficient for most people that use REST API, and you can find all of the documentations for REST API on rest.ensemble.org. And a few words about REST API, what it is and is not. So it provides HTTP access to Ensemble's data, and it is a stable service, which means that it doesn't change very much. It's limited by network latency, which means that the network speed is going to affect it. It's read-only, which means that you're not allowed to edit our database. You only have read access, and it's also versioned in archives. There are no mirrors, so you might know that we have Ensemble mirrors for US East and US East and Asia. It's basically the same sites, but we've got mirrors depending on the location. But we only have one REST server that is based here in the UK. Finally, it's not a hugely efficient data mining solution, so it's not as fast as direct as with MySQL or FTP side, and it provides incomplete coverage of what's available there in the database. And what is an endpoint? So here on my slide, I have a proper definition, but in a more human-friendly language, endpoint is a particular output that you can get given a particular input. So it's a function that interacts with our database. And you can find the full documentation of all the endpoints on rest.ensemble.org. And this documentation will list all the endpoints that are grouped by function. So they're grouped by the output, and you can find all of the required parameters for each endpoint and also the optional parameters on how to use them and the different programming languages on the pages as well. So I am going to jump out of my presentation, and we're going to look at rest.ensemble.org. So if you just go to rest.ensemble.org, I'll paste this in the chat as well. You'll see that we have all of our Ensemble REST API endpoints over here. And as I mentioned, they are grouped by function. So we have these endpoints over here for comparative genomics, cross-reference, information, and then we've got some linkage disequilibrium, lookup, mapping, and so on. The easiest way to find the endpoint that you're looking for would be by Control F, and then you can look for an endpoint. So for example, I'm going to look for the lookup endpoint or the lookup function. So I've got all the endpoints that are within this category. And I'm going to look at this first endpoint that says get lookup/ID. And this is the endpoint that you can use to find the species and database for a single identifier, which could be a gene, transcript, or a protein. So I'm going to click on this. And this is going to take me to this endpoint page. So this is our endpoint, which is lookup ID. The method is get. So there are different methods. So these are HTTP methods. We're going to be focusing on get mostly, but if you go through the Python notebook, you'll also see that we have post methods as well. But just for simplicity today, I'm only going to focus on this get methods. So our endpoint is get lookup/ID. Then anything after the colon is going to be your required parameter. In this case, it's ID. So our required parameter is the ID, and it has to be an Ensemble stable ID. And you have information about this just over here under parameters where it says required. So our required parameter is ID. The type is string. and the description needs to be the Ensembl stable ID. And here we have some example values of what these IDs could look like. Then we also have some optional parameters that you can use to extend your output. So you can see we have quite a few over here that you can choose from. Again, we have their name, we have their type, we have the description on what, how these optional parameters can extend your output. And then we also have some example values. Then if I scroll further down, you'll notice that we have some example requests. So we have our URL over here using this endpoint. And then we also have our examples of what the code in Perl would look like for this endpoint in Python or Ruby, Java, R, curl, or wget. I'm going to click on this this URL over here that says lookup/ID. We have our stable ID. And then after the question mark, it says content type equals application JSON. So I'm just going to open it in a new tab. And if you look at this URL, you'll see that we have our server, which is rest.ensembl.org. And then after the slash, we have our endpoint, which is lookup/ID. Then we have our required parameter, which is our Ensembl stable ID. And then after the question mark, you can add your optional parameters. So after the question mark, one of our optional parameters over here is that we want the content type to be application JSON. So that's just the format of your output. And this is the output that you get over here. In Google Chrome, if you're using this URL in Google Chrome, then the JSON output that you get is not super friendly to read. But if I just actually, I'm going to open Firefox. And I'll share my screen in a moment. So let me just go back and share my screen again. Okay, Batul, can I just check if you can see my screen?
I don't see it. I just see you.
Okay, let me just try again. Nope. Okay. I think it's not allowing me to share Google Chrome. Sorry, Firefox. So I'll just stay with Google Chrome. But if you just to point out here that you can copy this URL, and if you paste it in Firefox, then you'll get a nicely formatted JSON output with different colorations. That's just a lot easier to read. But if you just look at this output as well, so we provided our Ensembl stable ID here, and you can see that in our output now, we have information on the species that this stable ID is associated with, information on some canonical transcript, what gene it is, the start position of this gene, the end position of the gene, and so on. I mentioned before that you can always extend your output by adding optional parameters. So if you look at these optional parameters over here, I'm going to use this expand optional parameter, which is a Boolean type. So it's sort of like it has this, if it's set to 0, then it means that it's turned off. And if it's set to 1, that it means that this optional parameters is turned on. And you can use this optional parameter to expand the search to include any connected features. So for example, if you're searching, if you're looking up using this endpoint, looking up a gene stable ID, then all of its transcripts and translations and exons will be returned as well if you use this expand parameter. So I'm going to do that with our URL query. So right now, I only have the basic information, but I'm going to add another optional parameter. So I can do that by having a semicolon, and then I'll say expand equals 1. And if now I enter and look for it, then you can see that my JSON output is a lot larger because now it's going to contain the information on various different transcripts associated with this gene ID, all the translations and the exons as well. Let's go back to my presentation. So I've just endpoint documentation looks like. So again, the required parameters are indicated by this colon over here. So in the documentation, you'll see that that is something which you have to replace with your actual input. And then there is a list of optional parameters which can be applied. So you can add them to your URL and they follow this format. So there is a parameter name equals the desired option, and then this can be added after the question mark. So optional parameters will be added after the question mark, and you can add various different optional parameters. You just have to divide them by a semicolon. And then we have some sample code in various different languages that you can use. So this is something that I've already covered. So making a REST call in your browser is probably the easiest way. All you have to do is put your URL in your browser, and this can be used as a quick lookup. So you can test your URL to see whether they work, whether you've included the correct parameters, or what the output would look like. But you probably won't be using the browser calls very often, as you're more likely to be wanting to have your input in a file rather than in the browser. However, here we have an example of a browser call which you could be using quite often, as it is quite handy. So this is the ping endpoint, which confirms that you have a connection to our database. So here I have the URL for this particular call. So we're starting with rest.ensemble.org/info/ping. So this is our endpoint, which is info/ping. And then after the question mark, we have our optional parameter, which is our content type set to application JSON. So let's just try this in the browser. So I'm just going to copy this. I'm going to open it in a new tab. So again, I have my server over here. Nope. Let me just copy this again and let's try. I was failing at copying it, sorry. Okay, let's try again. There you go. So you can see we have our server over here, which is rest.ensemble.org. Then we have our endpoint, which is info/ping, and then our content type set to application JSON. And you can see that this query has returned ping 1, which means that we have a connection, everything is good. If you get anything other than 1, then it means most likely you don't have a connection to the database and our servers are down. And here we have an example of requesting a gene using the gene stable ID, which is just what I've shown you in the browser as well. So you can see that we have you have to construct your URL. So we have our server over here. Then we have our endpoint, which is lookup/ID. We have our required parameter, which is the ensemble stable ID. And then after the question mark, we have our optional parameter, which is content type set to application JSON. And this is what the nice formatted JSON output would look like if you have a JSON sort of plugin in your browser, or if you're using Firefox. So you can see that we have this returned JSON. So this is the structured data type that is composed of keys and value data pairs. So you could never just assume that your request ran successfully and the server uses HTTP code, HTTP status code to signal the request outcome. So for example, if your client makes a request for something that does not exist, so you either made a typo in the endpoint, So you're using an endpoint that doesn't exist, or you're requesting data of a certain ID that doesn't exist. Depending on the problem, the server is going to return a status code. So in this case, I've used this code 404, which means that URL was badly formatted. So basically this endpoint doesn't exist. And here in my slide, I have many other different status codes. So for example, 200 means that your request was successful. And everything else other than 200 means that something went wrong. So 400 means that your parameter was wrong. You asked for a gene stable ID, which is not found in the database. 403 means that you have made too many requests. So we do have some rate limits. We have a number of a quota for a number of requests that you can make per second. So if you've exceeded that, we will temporarily block you. So you'll just have to wait and retry again. Then we have 404, which means that the request was badly formatted. So most likely there was a typo in the URL. Then there is more. So everything starting with 5, for example, 503 means it's most likely our fault, which means that the server is down. And you can find the full list of all status codes in our GitHub in the link below on my slide. Right, so that was the introduction to REST API and how you can make the different URL calls. What we're going to do now is some exercises. So we have 3 over here. So the first one has asked you to find an endpoint which you can use to look up information about a gene using its symbol. So I just showed you in the demo how you can look up information about a gene using its ID. But now you have to use a different endpoint which requires symbol. Then you'll need to create a URL to find information about the gene, which is ESPN in human. So we have the gene symbol over here. And then the third part has asked you to include information about the transcripts. So if I go back to rest.ensemble.org, let me just type it here. You can see that we have all of our endpoints here. Again, if I look up for the lookup endpoint, you could just search the page using Control F. So this is the endpoint that I just used, which is get lookup/ID, because I was looking for the gene information using the ID. You'll need to use get lookup/symbol because the exercise has asked for symbol. So in this case, this endpoint has 2 required parameters. As you can see, there's colon and species, and then we have colon and symbol, because if you remember, gene symbols can be ambiguous. So the ESPN gene that the exercise has mentioned can also be found in zebrafish or mouse, as well as human, and maybe other species as well. So this is why it's important that if you're talking about a gene symbol, you also provide the species. So I'll give you about 20 minutes to do this exercise. And then if there are any questions, please do let me know. How are people doing with the exercises? Are there any questions? I'm getting a few thumbs up. Does anyone need more time? Do you need me to go through the exercise? Okay, so what I will do is I'm going to go through the exercise and then I'll move on to the next module. And then yep, and then we can have some time to spend on the on some scripting and using the Jupyter notebooks. So the exercise asked us to find an endpoint which you can use to look up information about a gene using its symbol, and then to create a URL to find information about the gene ESPN in humans. So ESPN is a gene symbol. And then to expand your results to include information about transcripts. So I'm going to go to rest.ensemble.org. And then from here, I'm going to search for the lookup endpoint. So we've got a few endpoints over here. So we've got get lookup/id. We've got post lookup/id. So post is just another HTTP method, and it can be used for bulk queries. However, we only have one gene symbol. So we'll be using the get method. And then this is get lookup/symbol/species/symbol. So let's open this in a new tab. So as you can see that we have our endpoint over here. And then we have two required parameters, which is species and symbol. Underneath, we have some information about these required parameters. So the description is given over here, and then example values as well. So it can either be the scientific name, or you can even provide the common name, and then you'll have to provide the gene symbol. Then we have some optional parameters over here, and then we have some example requests. So let's look at this example URL that we have. So you can see that we have rest.ensemble.org, which is our server. We have our endpoint, which is lookup/symbol. And then our first required parameter, which is species, and this is Homo sapiens in this case. And then in this example, the gene symbol is BRCA2, but I'm going to change this gene symbol from BRCA2 to ESPN, which is what the exercise has asked for. And then after the question mark, I have my optional parameter, which is set to content type application JSON. And if I click enter, I have my JSON output over here, giving me information about the ESPN gene. And then the third part of the exercise asked to expand the results to include information about the transcripts. So if I go back to my URL, so this is just a simple URL, or a simple, just one endpoint that I've used over here. But I can expand this endpoint to include information about exons and transcripts. So again, as I've mentioned before, that after the question mark comes your optional parameters. So one of our optional parameter is content type set to application JSON. This is just that what format you would like your output in, and we've said JSON. Then after the semicolons, I'm going to add another optional parameter, which is expand. And this is a Boolean parameter, so I'm going to set it to 1. And now if I click enter, it's going to give me lots more results about all the transcripts and translations and exons that are associated with this ESPN gene. So I hope that people were able to follow. And if you have any questions, please do let me know in the living document. And now let's move on to our module, which is scripting around REST API calls. So scripting around REST API calls is very useful, as you can extract specific bits of data from your call. You can output it in whatever format you would like. You can link together calls for more complicated queries, and you can integrate queries into larger pipelines. The great advantage of REST API is that it is designed to be accessed using any programming language. So you can make the calls and decode them within any script. In this course, everything is going to be in Python or R. If you go through the Jupyter notebooks, they're both in Python or R. But as I've showed you in the different pages for the endpoints, you can also use other languages such as Perl or Java and so on. For the Python modules, there are packages and modules that you will need alongside the standard Python or R for using the REST API calls. And these are the same for any REST APIs that you might want to use. So for Python, you'll need to have this request package. In the notebook, or these Jupyter notebooks that I've shared with you, this is already set up, so you don't need to worry about it. But if you're setting up your own Python installation, then this is something that you would need to set up. Then we will also be using the JSON package which ships most as standard with most Python installations, and then also Pretty Print. So it has its pprint to print your JSON in an easy to read way, and we'll be making use of Pretty Print quite a lot. If you're using R for the requests, it is the HTTR library and the JSON Lite package, and both of these come as standard with R. So now if we look at how to request information about a gene by using its ID. So we've got our server over here, then we've got our endpoint just over here, and this is the required parameter. And then after the question mark, we have our optional parameter. So what we recommend, which is good coding practice, is to make a string of the server and make a string of the extension. So the way the request package works is that you would put request.get, which is just over here, and then you put your full REST URL. So we have put it as making the server and extension as separate objects. And then we're combining them together over here. So we have server plus ext, which is our extension or our endpoint. Or you could put the whole URL in here if you'd like. And we've said that we want to use JSON, so we've put the headers in there, and then we have done a pretty print of this object, which is called R. In R, you would want to make sure that you're getting the libraries for HTTR and JSON Lite. And in this case, we use, again, we've got our, oops, sorry, too quick. We've got our server here, we've got our extension over here, and then I've created the object R, and then we are pasting the server and the extension together. I've put my content type as application JSON in except brackets, and then you can print command by just saying the name of the thing. So in this case, it's R. As I mentioned before, that your returned content types can be specified in the header as except. So you'll need to use content type in your URLs, and we've been using these as JSON. And all of the endpoint documentation pages are going to list the allowed content types. So over here, you can see that we have these different response formats. So the various different response formats, depending on the endpoint that you're using, could allow for FASTA, JSON, sequence, XML, text, YAML, or also JSONP. And you can find a full list of all of these on my on the link on my slide. So we also really recommend error handling or error checking, and we recommend using a, sorry, here it is. And we recommend using a command in all of your scripts to say, if the request didn't work, then tell me what the response code was. So you don't necessarily want to get a response code every time because your code has worked. You want to go on and you want to do the next step. However, if it hasn't worked, then you would like to know why it doesn't work. So in this example, we've got if not r.ok. So this gives me a response code of not 200. And it's got this raise for status. So it will only print the status if it's not 200. And if you remember from the status codes, if you get the 200 code, then it means that everything is working fine. But anything other than 200, it means that you might have an issue. And then in R, we've just got this stop for status. Then I've got some HTTP headers. So Ensemble REST uses a number of HTTP headers in REST to flag various parts of metadata to the user or to allow the user to influence what the REST API does. And just as I mentioned before, that we have these different content types that you can use or choose for your output. So we also have JSON parsers. So we've already said that we would that you can use these in Python or R installations to return the data as a structure. So in Python, if you put the name of your request or this r.json, that will give you the structure, and then you can pretty print it. So it's this pprint to make it display nicely. And then, as I mentioned in R, you do it as content, and then you can use prettify to print it in a neat structure. So JSON is basically a massive dictionary. So it's called a dictionary in Python and then data frame in R. And it has a lot of keys and values. So if you look at this on my slide, you can see that we've got this key over here that says source, and then a value. We've got an object type like gene, and it can be quite nested. So sometimes a key may contain other nested dictionary or list, and then this dictionary might contain another dictionary. So it can get quite massive and quite messy sometimes. So you need to look at the JSON to work out what keys you need, and you can cycle through these keys in the dictionary using the for loop. And you can find out more information about the for loop if you go through your Jupyter notebooks later on. So we've also written a helper function, which makes your life a lot easier. So it calls the request with the server extension and the content type that you give it. It gets the status and it decodes the JSON if you've used that as your content type. If you've used a different content type, then it will just return it to you as plain text. So it's really useful if you write really complicated scripts. So you can just stick the helper function at the beginning of your script and then use it every time you want to fetch an endpoint. And it will work with any REST API. So not just the Ensemble one, it will work with everything. And I'll show you what it looks like in Python. So we put define first, this function that we've called fetch endpoint, which is made up of the server, the request, and content type. You have to create these objects and then you need to put them in. So now it runs the request option. It raises the status and it exits the script if you get a failed query. So this means that you won't get stuck if things need to feed into each other. And you've said that the content type is JSON. So it will decode your JSON for you. Otherwise, it's going to give it to you as plain text. In R, it's pretty much the same thing. So we've got this fetch endpoint as a function with the parameters. We've got our server, request, and content type. So it runs the get request, that's just over here by pasting all of these objects together, and it does a stop for status, which is the error handling, and it also decodes the JSON if it needs to. Otherwise, if your content type is anything other than JSON, then it's going to return it as text. Okay, and now we are on our second set of exercises. So again, we are following on from the first exercise. We're still going to look at the ESPN gene, but right now, instead of creating a URL, you're asked to write a script to look up information about the gene called ESPN in human and then print the results in JSON. So in this case, again, you'll be using the same endpoint, which is lookup/symbol. And we're going to be using our Jupyter notebooks. So you can see that we have information over here if you scroll down about the helper function. So we've got our error handling over here, and then this is your helper function. And over here, you've got space to write your code. Where it says exercise 2, you can just write your code over here. I'm going to give you another 20 minutes for you to go through the notebook a little bit and also look into how you're going to write your script. And then after that, if there are any questions, please do write them in the living document or even the chat if you'd like. Okay, so I hope that was enough time for you to go through the exercise and write your own code. What I will do now is I will just demo it onto the Jupyter Notebook as well, and then we're going to close the session for today. So let's go to our Jupyter Notebooks. I'll demo it in Python. So the exercise asked us to write a script to look up the gene called ESPN in human, and then print the results in JSON. So I'm just going to unhide the answer here, and then we can just go through it. So what we've done here is that we've got our modules that are needed for the script. So as I mentioned, that we will need the request module and then also the pretty print. Then we have got our so if I just scroll down a bit, so we have our endpoint over here. So this is fetch endpoint, and we've got server, request, and content type here. So this is our object. So we're calling our package, which is request.get, and then we're pasting together the server and the request, and our headers are accept, which is content type. Then we've got our helper function over here. So we've got all of our the race4 status. So this is the error handling. And then we've also said to decode the JSON. So if our content type is in JSON, then give the output in JSON. If anything else, then please return in text. Then we've defined our gene, which is gene name ESPN. We've got our server over here, which is rest.ensemble.org, and then our extension. So this is our REST query, which is lookup/symbol. We've got our required parameter, and then we're pasting in our gene name. We've defined our content type, which is application JSON, and then we're going to submit our query. And if I click on play over here, it's going to run the code for me, and it's going to give me the output in a few seconds, which is just over here. So we've got our output over here for the ESPN gene. So you can see that we've got the biotype for this gene. We've got our canonical transcript that is associated with the gene, the display name, the description, the start position, the end position, source, and so on. You can see that I'm only going to cover the first two modules for today, but as I've mentioned, that these Jupyter Notebooks are yours to keep now. So you can look at the further examples that we've got here. So the next example is on how you can fetch specific data points from your JSON output, because usually you're not going to require the entire JSON output. You'll usually want just one single data point from your output. Then we've also got, if I keep scrolling down, we've also got examples over here on other content types. So I've just shown you the JSON content type, but obviously if you're working with sequences or if you want information on gene trees, then it will have different content types. And then you can find all of these within this examples in module 4. Then we have another example on how you can link different queries together, so different endpoints together. And then finally, we've also got a module on post queries. So these are bulk REST API calls. So as I mentioned, get query is only for one item at a time, whereas post, using the post method, you can do multiple inputs at once. With this, I would like to thank all of you for your attention. Before we sign off for today, I would really appreciate if you could give your feedback. So you can find the feedback survey on the training page. So this is just over here. If you go to the training page, this is the link to the feedback survey. I'll also share the link with you in the chat. So I'll give about 5 minutes for you to fill the feedback survey out. And then you can also get a certificate of attendance. And then after that, we'll come back and I'll do a quick wrap up. Okay. So I'd like to just wrap up now. To start with the citation. So if you use Ensemble, please do not forget to cite us, as it would help us realize who uses our resources and how. And we've spoken about Ensemble Genome Browser yesterday. And today we covered REST APIs. So the Ensemble browser sites would include the main website. Then we also have GRCH37 that is dedicated to the previous human genome assembly. We have Ensemble Genomes that is dedicated to non-vertebrate taxa such as fungi, plants, bacteria, metazoa, and protists. And then we also have our archives, which are previous versions of Ensemble stored in time for up to 5 years. We've also spoken about Biomart, which is a data mining tool. And then most importantly, Ensemble's database is open source, so it's open to everyone, and we give you access programmatically as well via Perl API, REST API, and MySQL. We also give you access to our whole genome flat files via our FTP sites. If you have enjoyed the course, then please do recommend us to your friends. So I sort of did a more condensed course yesterday and today based around the browser and REST API, but we also do a day-long course. It can be about like 8 hours, covering quite comprehensively about the different data types that we have. So do recommend us to your friends and colleagues. And if you would like to host a Ensemble Browser workshop or REST API workshop at your own institutes, then please do get in touch with us on helpdesk@ensemble.org. There is a lot of help and documentation that is available on using Ensemble, and also various other bioinformatics resources, and you can find them on the EBI training pages and also on various different tutorials on Ensemble and our YouTube channel. If you still have questions, then please do get in touch with us on helpdesk@ensemble.org. You can also join our public mailing lists at dev@ensemble.org or announce@ensemble.org to keep up to date with any new jobs, internships, or any new updates that have been brought about with new releases. You can also follow us on Facebook and Twitter. And with this, I would like to thank all of you for your attention, and also the team and our funding bodies that make this entire process possible.